We are now moving into post-harvest treatments of post-harvest rots with a focus on hot water treatments. And before we look at hot water treatments, we really ought to look at the rots which we hope to control, because we need to understand, again, as I said in my introduction, we need to understand the biology of these fungi in order to optimize the way in which the technique might work. This is a picture going back to Peter Maxine's PhD, and it just shows the diversity of rots which you might have in a single orchard. In this case, a veteran orchard of fruit rot research. It is the Ingrid Marie orchard here at Orslev. And it has been left untreated after flowering for many, many years, and it harbors the greatest possible diversity of storage rots. So it really should be put under some kind of monumental protection. There should be a protection order on it that it can't be uprooted ever. Anyway, looking at storage rots, this is a slide which my predecessor, Dr. Gerd Palm, compiled, and it shows the percentages of different fruit rots in different crops of northern Germany in the late 1990s and early 2000s. Well, we can see at one glance that the dark blue color seems to predominate. That's one particular fungus which is very common in northern Germany. But if we focus on two orchards harvested the same year, Gloucester and Elster, 2001, we see an almost mutually exclusive spectrum of fruit rots. So there can be a variation in the occurrence of fruit rots in space over a production area such as the northern German Altes Land. There can be variations also in time. And this is a compilation of data which I gathered from the literature. There was a paper written in our regional journal in 1958, which shows that in those days, Neofabrea species, they are the ones which we formerly called Gliosporium, were dominant at the time. And that continued until and including 1968. In 1968, we had a legendary storage rot year in which almost 90% of the fruits which the farmers put into storage were rotted when they took them out again. And Neofabrea species, these so-called Gliosporium fungi, were to blame. In 1969, a new fungicide, Benomil, was introduced into apple production. Benomil is a benzimidazole fungicide and had an extremely high efficacy if applied just before harvest against these Neofabrea rots. And as a consequence, they all but disappeared from the spectrum of rots in the next few decades. And this is an example of that time when other fungi took over, for instance, Neonectria, which is the cause of apple canker. But then from the mid-1990s onwards, Gerd Palm observed how Neofabrea species were gradually clawing their way back to regain their former position of dominance. And he suspected that fungicide resistance of these fungi against the Benomil compound or other benzimidazoles was to blame, but he couldn't prove it. Well, I could because I was familiar with the microbiological methods, and so the first task he set me when I joined his department in 2006 was, do go and have a look whether there's resistance in these storage rots. And this shows inhibition curves of Neofabrea perennans. These are three sensitive isolates, and you can see that at below 0.1 parts per million, the growth of these sensitive isolates is completely inhibited. Here we have a partial resistance where we need concentrations which are more than tenfold higher to give rise to an equivalent level of inhibition. And then there are yet other isolates which grow irrespective of the concentration of this compound, in this case thiophanate methyl, which is related to Benomil. I presume you can grow this on crystalline Benomil and it will still grow. And these two types of resistance, the partial resistance and the full resistance, can be correlated to different mutations in the beta-tubulin gene, which encodes the molecular target for this fungicide. So it attaches itself to beta-tubulin, which is a cellular component of fungi, and then it inactivates it, and so it inhibits the fungus. And in these two mutations, the docking site is slightly modified so that the fungicide loses its efficacy. And I was able to show that these types of resistance are spread all over northern Germany and other parts of Germany as well. So presumably people have been spraying with these fungicides for years without them being actually effective. And as a consequence, a year later in 2008, these fungicides were more or less discontinued from use across Germany. So these are the three species which cause the Gliosporium type rots They used to be called Gliosporium. This one, Gliosporium perennans, now it's called Neofabria perennans. This one here, Neofabria alba, formerly Gliosporium album. And this one is particularly interesting for two reasons. Firstly, it is important in Denmark. And secondly, it used to be misidentified across Europe for decades. It was just given the wrong name, and my colleague Arne Stensvand from Bioforsk Norway found out about this. And that just shows the fact that all plant pathologists got it wrong for decades. That just shows the extent to which one copies the writings of others without actually checking for himself. And because Gliosporium, or Coleotricum acutatum as it is now called, has a totally different response to fungicides from Coleotricum gliosporioides, which would be the current name of Gliosporium fructigenum, this is important. So these three species are the most important, in their totality, the most important storage rots in northwestern Europe. In organic orchards or in untreated orchards, you might get anything from 10 to 25% in organic, above 25% in untreated orchards of the crop, which can be lost during storage to these rots. As I said, in 1968, over 90% in conventional production. These storage rots are more common in the second and third pickings, second and third harvests than the first harvest. So the fruits you leave longest on the tree are the ones that are most strongly affected by these rots. And their distribution seems to differ across northwestern Europe. It does seem to be a northern German speciality to have Neofabria perennans as the dominant of these three species. Both in Denmark and in southern Germany, Neofabria perennans is quite rare. In contrast, in these two regions, Neofabria alba is the most common storage rot. In northern Germany, only on certain varieties. It's very strange, we don't have an explanation for it, but that seems to be the case, year in, year out. Coleotricum acutatum is a fungus which is of very, very low importance, generally speaking, in northern Germany and southern Germany, but it can be very common in Denmark, as Marianne has mentioned. So all three species which I've mentioned can be associated with fruit mummies, and my survey has shown that. And the question, of course, is, if we remove fruit mummies, do we get an effect on storage rots? Unfortunately, we don't. At least no significant effect. And that's probably because these storage rot fungi are present also on other organs within apple trees. With the Neofabria species alba and perennans, I was able to show that they can colonize cut ends of twigs. So pruning during the summer, for instance, will generate wounds, and these fungi can infect these wounds, can cause a very local, slow-growing canker, and there are conidiomata produced on the surface, and the spores can be washed down and can presumably infect apples just before harvest. And there are many other habitats where these fungi may be found. We don't know enough about these habitats to recommend measures. So this is the share, this blue section here is the share which is generally occupied by Neofabria species and by Coleotricum acutatum. In this case, this is again our veritable Ingrid Marie orchard occupied by Neofabria alba. But as we can see, about a third of all the fruit rots can be contributed by other species. And this is, of course, where mycology comes in. It's very interesting to have a look at some of these other fungi also causing fruit rots. I'm going to discuss most of them in the next few slides. Let's start with this one, and you will have seen it in the boxes which are on display today, botrytis grey mold. Botrytis is a very variable occurrence, and we can find at least one reason why that should be so. The fungus is able to spread from fruit to fruit during storage. So by just putting one infected fruit into your storage bins, you might end up with a nugget or cluster of 10 fruits as you take the fruits out again, 10 infected fruits as you take the boxes out again from long-term storage. And it's quite rare that a fungus can spread from fruit to fruit. Botrytis is a very big issue, to me anyway, because I conduct research on it in another context, and that is the context of fungicide resistance. looking at, particularly in strawberries, botrytis is the major cause of fruit rot in northern Germany and also in Denmark. And we can see that the fungus has developed resistance to all fungicides which are currently in use, both partial resistance shown in yellow and full resistance shown in red. And the level of resistance is quite high, and many isolates actually have a multiple resistance to all fungicides which we can use, both in Denmark and in northern Germany. The same applies to isolates from cherries, and there is really no difference in that isolates both from soft fruits such as strawberries and cherries seem to be able to develop resistance to any fungicide we care to throw at them. But on apples, it's a totally different matter. Apple isolates are virtually never resistant to any fungicide, and those few bits of percentages of fungicide resistance we see are usually due to other isolates which have moved in. And to cut a very long story short, we are beginning to find, and this is a collaboration with the University of Kaiserslautern, we're beginning to find that the apple botrytis fungus is an altogether different species from those infecting cherries and strawberries. And because it has a very much reduced capacity to develop fungicide resistance, management options are also different. Another fungus which causes storage rots relatively frequently and which we can also see and recognize easily enough is Neonectria ditissima. It used to be called Nectria galligina, and it is the apple canker fungus. And I've included a few canker twigs there in the display. This fungus is special because it can infect at different periods, at separate periods of the apple's lifetime. It can infect during flowering and again before harvest. And both types of infection correlate with apple canker. So trees which have a lot of canker are also more likely to develop fruit rot. And of course, by pruning canker regularly, you will also control this kind of fruit rot quite effectively. This slide shows the kind of symptoms you can see with Neonectria. This is the blossom end rot. These are infections at flowering, which then manifest themselves during the growing phase of the apples. And usually you can see it quite clearly from the outside. But sometimes, if the fungus gets into the flower canal towards the core of the apple fruit, you won't see it from the outside. And then if you take one of these fruits and you harvest them and put them into storage, then of course you get a storage rot. The second phase of infections takes place just before harvest, and these infections are present on or in the fruit skin as latent infections, and then they break out like a normal storage rot, like Phacedopichnis and also like Neofabrea. The key feature of Neonectria rots is that you can separate the diseased from the healthy tissue very easily by just taking a spoon or spatula. So if you like, you can spoon out the diseased tissue. I don't know whether anyone would like that, but anyway, it is a diagnostic feature. This graph shows that fungi don't read publications which have been written about them. Because for decades, the accepted wisdom was that blossom end rots, for example Neonectria, infect the flowers after or at earliest during petal fall. And Hinrich Holthus and I have been conducting experiments for several years now where we artificially inoculated apple trees, and we find each year that the full bloom period is actually the time when these infections take place. And that has an obvious bearing on fungicide recommendations. So you have to bring forward the spray by about 2 weeks. We have not been able yet to artificially infect fruits or flowers with botrytis, incidentally. So we have to work on the technique. This slide shows a monitoring of blossom end rots in Germany, in northern Germany, for the last 7 years, a survey. And you can see that botrytis, shown in grey here, and Neonectria, shown in blue, are the dominant species. But the dominance of either varies from year to year. And you can correlate that with the weather during flowering. So if you have a very dry period during flowering, botrytis will be the major blossom end rot. If you have wet periods during flowering, Nectria will be the main cause. Of course, that's the time when we also get a lot of apple scab. And you can tell from this diagram when I took over the scab warning, the responsibility for scab warning. Of course, it was in 2013. So that's when we got the wet flowering periods. Part 2 of our Spot the Rot contest. You can have a guess as to which of these apples just before harvest is actually infected by a rot. It's quite easy to see, I think. This prematurely ripened fruit here at the bottom is the one, and If you get a premature ripening of apple fruits, usually this is associated with an infection of the apple seeds by a fungus. And this is called a core rot. And that's often due to Fusarium, more precisely Fusarium avanaceum. If a fruit, such as the one I've just shown, with a rotten core but looking still moderately intact from the outside, is put into storage, then of course you get a storage rot, which has quite nice color, pigmentation, but that's little comfort to the affected fruit farmer. It is a fairly aggressive rot, and it is particularly dangerous because Fusarium avanaceum can produce mycotoxins. So if you put fruits such as this one here to juice production, then you might get detectable levels of mycotoxins in your juice. Monilia is another fungus causing storage rots, and it is also a pre-harvest rot-causing fungus. It infects through wounds, such as those caused by hailstones, or in this case cracking of the fruit due to sunburn, and the fungus can be spread by flies. Now this fungus does not have any latency period, so as soon as it infects a fruit, the brown rot will break out. And therefore, consequently, if you put fruits which are being infected at harvest, for example, if you have forgotten to cut your fingernails when fruit picking and so on, if you cause wounds and you introduce Monilia during harvest into your fruits, then it will break out very soon. It will be the first fruit rot to be visible in storage. And that tends to lead to an overstatement, to an exaggeration of the importance of this fruit rot because it's so commonly visible. And visible is the first species to become visible. And that's quite in contrast with this rot here, Penicillium expansum. This is the only fungus which actually exclusively infects apples during storage. So you cannot show its presence, hardly ever find it on harvested fruits, but it will gradually build up in the air circulation system during long-term storage. These spores are very small and they are airborne, and gradually you have an inoculum build up and more fruits become infected. It's a second way by which the fungus can spread is the grading after you've finished your storage period. So boxes are usually dumped in water, called bin dumping method, and then you grade the fruits in a water grading machine according to size or color or whatever, and you sift out the rotten fruits. If a single fruit as this, such as this, can harbor 6.9 billion spores, and we found that you need about 1 million spores per liter grading water to infect any fresh wound which is available. So just one single fruit like this is sufficient to contaminate 6,900 liters of bin dumping water such that any wound will be infected by this fungus. So that's a second very effective way. This is a kind of summary in which I've tried to sort the various fruit rots according to the time when the symptoms become visible on the apple fruits. And you can see that fruit rots can become visible early during growth. They can be visible at harvest. For example, Diplodia, our black rot fungus, is visible during the growth phase well before harvest. The Fusarium rot is present at harvest. Many of the storage rots, as the name suggests, emerge during storage. And there are some fungi, and that's a nice little exercise. I would love to follow up this a little bit more, but I really should focus on more relevant things, so I'm not going to do it. But of course there's a temptation, which we mentioned earlier. These are rots which are present only on sections or on patches of the fruit which have been killed or destroyed by other means. For instance, by excessive heat treatment, giving rise to heat scald, or by cold injury. So dead regions of fruits often show fungi colonizing these, which you wouldn't normally see. So we have a kind of cryptic group of storage rot fungi, which you never see on healthy fruits. That'd be a nice little PhD topic. Fruit rots which are present only on dead tissue. I don't know whether we'll get funding from farming associations for that, but we could try. It's worth it an effort. So the whole fruit rot section, including the pre-harvest rot Diplodia, has been worked on by several colleagues, including myself, of course, and I'm happy to acknowledge their names here. Gerd Palm and his chief technician, Petra Kruse from the Esterborg Center, Hinrich Holthusen, who joined us, our department more recently, Gesche Quast, who as a trainee technician was working on Diplodia, and there have been several points of collaboration with Aarhus University, with the Department of Food Science here, Peter Maxine during his PhD, Annette and Stig Sørensen, and of course also Marianne Berthelsen. So thank you all very much for this collaboration. And I'm now moving on to the last topic of today's talk, that's hot water treatments of apples. This slide shows how complicated the machine is which has to be used for hot water dipping. Hot water treatment isn't really a new concept. In fact, it's a bit of an old hat. Burchill in 1964 very aptly described the key principles of hot water treatments. But Burchill was unfortunate in performing excellent work at the wrong time. Soon after his publication, the benzimidazole were released, an effective fungicide was available, and hot water treatments sank into oblivion. Gerd Palm just did examine hot water treatments, and he found that the current standard method of 3-minute dipping at, say, 53 degrees was more effective than repeated sprays with fungicides before harvest. And there has been a certain restoration of interest in hot water treatments as more and more farms turned organic and were looking for new approaches to control storage rots, which were the major problem, one of the major problems in organic farming. But unfortunately, the method of hot water treatments has failed to live up to its true potential. And there are several reasons for that. First of all, as I've shown in the preceding slide, specialized treatment, specialized equipment is necessary, and that's very expensive. So a few farmers in northern Germany do perform hot water treatments, but you can number them certainly on 2 hands, unless you've pruned off several fingers due to accidents. Hot water treatment has to be performed in its current shape during the peak work time of harvest. So really, when a farmer needs to have all hands on deck for picking fruits, he would need extra men for dipping fruits. And that's not considered possible by many. A third point, which is also important, is that there is an absolutely appalling CO2 footprint associated with hot water treatments. And organic farmers who praise themselves as being particularly environmentally friendly really have a bit of a difficulty in explaining why they should use 3 liters of fossil fuel per ton of dipped apples. And of course, that also gives rise to high costs. So these are all arguments against hot water treatment. For some reason, the method doesn't really seem to have caught on properly. And when Peter Maxine, who was working at the time in the Esterburg Center, came up and discussed the possibility of doing a PhD on hot water treatments, we decided that we would take a step backwards to work on the fundamentals of the method a bit more, to understand the fundamentals before we could hope perhaps to achieve a breakthrough in implementing it into practice. So one of the key questions we asked ourselves was, how does it actually work? The accepted knowledge is that heat kills fungal inoculum. So it is the direct effect of the heat during the dipping on the fungal spores which are on or in the apple that will kill, the heat will kill the spores, and therefore the rots can't develop. But we had certain doubts about this theory because we saw that Peter saw from his previous work that heat treatments always were most effective at temperatures just below those causing the first heat damage on the fruits, irrespective of the rot which was to be controlled. And we know that different fungi possess totally different heat sensitivities. So there didn't seem to be an obvious correlation. It was always the same temperature at which different fungi were controlled. The first step we did was to look at the actual, to quantify the heat sensitivity of different fungi, of their spores, presuming that spores are the most important source of inoculum. And we dipped the spore suspensions for 3 minutes in water of different temperatures. And you can see that there are massive differences in the heat sensitivity of these storage rot fungi. Some, Neofabrea species and Phacedia picnis, for instance, have a very low heat resistance. The temperature killing 50% of the spores is in the region of 39 to 40 degrees. Some are of intermediate heat resistance, Botrytis and Neonectria, for instance. And we have Penicillium expansum, the blue mold, which has a very high heat tolerance, and its spores are completely killed by temperatures in the 60s, in the lower 60s. So we used Penicillium expansum as a fungus for our further studies. And we were quite surprised initially when we took this temperature-resistant, this high-temperature fungus. We wounded fruits, inoculated them with Penicillium expansum, and we found that although the spores are killed at temperatures well above 55 degrees, yet hot water treatments at these temperatures here from 47 to 51 degrees were very effective in controlling artificially inoculated fruits. When Peter went on to dip fruits at higher temperatures, he found a resurgence of blue mold, so the fungus actually was able to cause infections at 54 or 56 degrees, but much less so at 51 or 49. And these results were totally incompatible with the notion of a direct effect of the heat on the fungus. But still, we were curious to find out how hot we actually needed to dip the apples to get the direct effect. So Peter conducted the experiment again with a wider range of temperatures, scalding his arms in the process. And that's what we need. We need people to make sacrifices in the course of science. And Peter resolutely did that. And we found the same picture again. By dipping at very low temperatures, there was very little effect. Then we got a trough of penicillium growth, i.e. the highest efficacies around 50 to 52 degrees. And then the blue mold came back. And only when you dip fruits at 70 degrees for 3 minutes do you actually find that you can eliminate Penicillium expansum entirely from these fruits. Of course, the apples were cooked. So this is not of practical relevance, but we have a proof of concept that this is the temperature which you need directly to kill Penicillium expansum. So quite obviously, this effect here wasn't due to this trough here at 52 degrees wasn't due to a direct effect of heat on the fungus. Well, so if heat hasn't got any effect on the fungus, it must have an effect on the apple itself. And if heat has an effect on apple fruits, then it might be possible to inoculate the fruits after dipping and to see this effect as well. So if the fruits are in some way immunized against or strengthened in their immune response against fruit rots, then maybe if we infect after the dipping, we would see a similar response. So it was poor Peter had to perform the whole experiment again, this time inoculating the fruits after dipping. And that's what he found. The expected result, with a bit of a difference which we can't fully explain, we can repeat these results even if the fruits were inoculated after dipping. If the fungus, in other words, was not exposed to the heat at all. The only difference, of course, is here at 70 degrees, and that's obvious. If the fungus hasn't experienced the temperature itself, it can't be killed directly by heat. So let's just summarize a few important points about the basics, about the mechanism of hot water treatment, the mode of action. The most effective temperature for hot water dipping doesn't correlate with the heat sensitivity of fungal spores, but it is always just below the temperature which causes the first bit of damage of the fruits. So you need to go as high as possible without actually damaging your fruits. This effect it can be shown to be present even when the infections by the fungus take place soon after dipping. And that has been shown to occur for different crops in the literature as well, not just by us for apples. Hot water treatment can be effective even against heat-tolerant fungi, and it is obviously effective via a stimulation of the immune system of the fruit. And this is a summary showing the responses of several fruit rot fungi based on actual results, but just shown as a scheme here. We've seen the red line for Penicillium expansum. If we take the very heat-sensitive fungi, such as Neofabrea, we would get a synergy of the heat shock effect on the fruit and a direct effect of the heat killing of the spores. But if we take those species of intermediate heat tolerance, such as Neonectria and Botrytis, you do get a certain resurgence of rot at temperatures causing the first fruit damage, and then it tails off as we get the direct heat inhibition of the fungus. So 3 different kinds of response are possible depending on the heat sensitivity of the fungus. What could be the physiological basis of heat shock? Well, having established that there is an effect on the apple, it was easy to get into the literature and to find that heat shock responses, of course, are well known in fruits. They have been characterized especially for citrus fruits. And sublethal temperatures can induce the transcription of genes which encode particular proteins These could be heat shock response proteins in general, and a subset of these are pathogenesis-related proteins. They are, for example, enzymes which can degrade fungal cell walls. So they respond to heat shock in being formed, in being synthesized. Or they could be biochemical pathways leading to the synthesis of low molecular weight compounds, phytoalexins. They are a kind of inhibitory substances produced by plants against fungi. We needn't worry about the details here, and I'm not going to go into further detail. Peter found two additional points of interest, one more theoretical and one of relevance to the fruit farmer. Point 5 is the one which is of interest to fruit farmers, because Peter could show that fruits could be stored for several weeks in the cold room or for several months under ultra-low oxygen conditions, and they are still competent to become immunized by heat shock. So you don't need to dip your fruits directly after harvest. You can actually store the fruits for a little while, and when the pickers have finished picking the fruits, you can still treat them with heat shock and then store them away again. And we found that ties in with what I said at the end of the previous section. Hot water treatments at super optimal conditions can increase fruit rots. If you kill the extreme surface of an apple and the fungus is still just underneath, it can actually grow better on the dead tissue which is then available. Of course, very interesting to use heat shock to bring out fungi which you normally wouldn't see. But anyway, I must resist this temptation. It's difficult. So if hot water treatments act on the fruit and not on the pathogen, we would expect this method to be capable of controlling a very wide range of fungi. And that was the second part of Peter's PhD. And this is again where our Ingrid Marie orchard at Orslev came in, because no orchard harbors such a marvelous diversity of rots as this one. And we wanted to work specifically on natural infections to show what hot water treatment could do for farmers. We didn't want artificially to inoculate our fruits. So we supplemented the Ingrid Marie orchard, the spectrum of fruit rots of which we knew, with apples we had smuggled across customs from across the border from northern Germany. And they were all treated, and we evaluated all these rots. Took us several days of great fun we had in the glass houses looking at fruit rots. And these are the results. This slide isn't rocket science, because it just confirms what we know from the literature. The two Neofabrea species forming Gliosporium type rot are very effectively controlled. The top row of this table shows the percentage of each rot in the apple crop after storage. So 25% of fruits of this batch here were rotted by Neofabrea perennans at the end of storage. And the large numbers printed in red and bold indicate the efficacy of hot water treatment, i.e. the percentage by which hot water treatment reduced these fruit rots. And we can see that there is a highly significant effect on Neofabrea alba and on perennans, and also, generally speaking, on Monilia fructigena. That had been known before. The next slide is new, and it extends the range of fruit rots that can be controlled effectively by hot water treatment. And these two here are very important for Danish fruit production. Phacedia pycnis washingtonensis is a fungus which is common in Denmark, and Coleotricum acutatum we've seen also to be important. This one here, Cladosporium, is less important, but anyway, there it is. Some fruit rots cannot safely be controlled by hot water treatments. And we have explanations for that based on what I've told you before about the biology of these fruit rot fungi. Neonectrioditissima, the apple canker fungus, as we have seen, can form blossom end rots, or it can form hidden core infections of apple fruits. And if these are put into storage, we can't really expect a hot water treatment of a fruit on the outside of the fruit to be effective against such a fungus. And the same applies to the core rot caused by Fusarium avanaceum. With Penicillium expansum, it's different. This fungus will infect months after the fruits have been heat treated and put into storage. So after such a long time, you wouldn't really expect the hot water treatment to be effective. So in other words, hot water treatment will not work against fungi which are separated from the hot water, either in space by being hidden within the fruit, or in time by infecting long after the hot water treatment has taken place. Or to put it in other words, a fungus is susceptible to control by hot water treatment if it is present if its infections or its inoculum are confined to the outermost fruit surfaces at harvest. Again, there has been extensive collaboration between the Esterbrook Center and Aarhus University, and I'm happy to acknowledge the people I've listed here: Stine Annette Sørensen, Marianne Bertelsen, Peter Maxine, and I would like to extend my thanks to the heads of our two institutions, Michelle Williams and Carsten Klopp, for permitting us to conduct this research and for encouraging our collaboration. Thank you very much for that, and I'm happy to see all of you, all of the people mentioned here, present today. Peter's PhD was quite tough for him and for me as well. We did manage it in the end, and he succeeded brilliantly, but we really had the feeling that enough was really enough, and we were quite relieved that it was all wrapped up nicely and the publications had been made, and just mentally speaking, we were sitting down in our armchair, putting up our feet and drinking a good pint of Danish ale, when our peace of mind was shattered by my colleague Hinrich Holthusen, who came up and asked whether he could also do a PhD on hot water treatment. And would I do a bit of supervision for him as well? After taking a few breaths and regaining our composure, we were thinking of ways in which such a project could work. And this project, which Hinrich is currently working on, or has just started working on, is based on some aspects of Peter's work which I haven't mentioned so far. Peter was also concerned with the question that if hot water actually stimulates the fruit rather than inhibiting the fungus, maybe it's possible to trigger this heat shock by reducing the time of exposure of the fruits. So he was experimenting with reduced hot water exposures from 3 minutes at 52 degrees to 25 seconds at 55 degrees. And he found a fairly high efficacy under those conditions as well. There are advantages, of course, of such a method, especially if you dip pre-cooled fruits. The shorter the exposure time, the less awful the CO2 footprint is likely to be. You can integrate shorter dipping times better into water grading lines. So you could, if you grade your fruits in your grading water, you could introduce a module through which those fruits which have to be hot water treated might pass. And you could perhaps even adapt it to integrated production, because although we have fungicides that can moderately control storage rots, of course the fungicide applications just before harvest are the ones most likely to give rise to residues in the product for sale. And we wanted to use and adapt a commercial machine for these purposes. We didn't want to develop a new machine, but we imported one from Israel, where it is used especially on citrus fruit production, and we wanted to try to adapt it as a prototype for apple production in northwestern Europe. And this is a photograph of an introductory seminar which we had in December 2014, December last year. We can see Peter and Hinrich here explaining how the machine works and showing it in operation, and I was particularly happy to see a major Danish contingency present at that meeting. It just shows the extent to which we interact and we exchange knowledge and also visits. This is all I want to say. I don't want to carry on talking about Hinrich's project, because it is not really appropriate to count your eggs before the chickens have hatched. I would, however, like to outline a few perspectives which I can see from my affiliation with Aarhus University. I feel deeply honored, and it is a great pleasure for me as well to be affiliated in this way. And I can see several points in which I might be able to make a contribution. First of all, I think our institutions are perfectly matched to each other, because we have complementary research resources. There's little duplication, but a lot of complementarity between the Årslev site here and the Esterbrook site. So we should harness our complementary resources to work more effectively on solving problems. And Merete's talk, which she's going to give in a minute, is a perfect example of something which is of great interest to us, but which we could never hope to do and wouldn't want to do at the Esterbrook Center, because we would just frazzle out too much in our core research activities. So it's nice to divide up the techniques and the approaches between two separate institutions. I'm very interested, of course, in contributing to joint supervision of PhD students. Hinrich is the second one from Aarhus University I'm co-supervising. And I'm also more than happy to train other students or technicians in mycological methods. It is obvious that if we join forces, we have a better chance of acquiring financial support for our institutions, which is very important in these times. I'm happy to contribute to teaching at AU Food, and we've discussed ways to do that in the shape of the master course during the break. And one thing I feel very strongly about is that regional research institutions should support regional advisory services. We do that in Northern Germany. We've been doing that for the last 80 years, backing up our consultancy service by regionally conducted research. And I think we should also, I'm also very happy to contribute to supporting Danish advisory colleagues with the work which I'm doing. In other words, we should generate practical knowledge with the aim to serve fruit production. I'm very happy to see representatives of the fruit farming, fruit farmers here, both from Northern Germany and from Denmark. And it is particularly important. Your feedback is particularly important to me because it serves as a kind of control mechanism as to the purpose and sensibility of what we do. Thank you all very much for your participation today, and well, thank you for your attention. Over to you, Merete.